Hey guys, it's me again, Sean Nugent here. Today I'm going to be explaining what type of an inheritance a two-trait cross is and how to solve a Punnett square problem based on one. Now before we move on, let's set a like goal of 10 likes on this video. I know I'm asking for a lot, but I know you guys can do it. Now let's jump right into the video. Let's start with the basics. What is a two-trait cross? Basically, a two-trait cross, other known as a dihybrid cross, is a genetic cross between individuals with different alleles for two gene loci of interest. Now, D in dihybrid actually stands for two in Greek. Interesting fact, the dihybrid cross was used by the geneticist Gregor Mendel, which helped him to formulate his law of independent assortment. Wait, stop, Sean. How am I supposed to tell the difference between a dihybrid cross Punnett square and a simple two-cross dominant recessive Punnett square? Now careful, this isn't your average dominant recessive two-trait Punnett square. In a dihybrid cross, the two separate traits undergo independent assortment, meaning that how one trait is inherited doesn't affect how the other trait is inherited. The dihybrid cross Punnett square also contains 16 squares as opposed to the 4 in the dominant recessive two-trait Punnett square. So now that we know what exactly a dihybrid cross is, let's see what it would look like as a Punnett square problem. On the left, I listed a key that shows the genotype and phenotype of each trait for a pea plant. Both of these traits show complete dominance, which means that if there is one dominant allele, the plant shows the homozygous phenotype. To solve this problem, we want to be able to show the cross between the traits. In this case, we will be crossing the heterozygous parents, so the parents are going to be big T little t, big R little r, and big T little t, big R little r. To make a dihybrid cross, we need a 4x4 Punnett square, which is a Punnett square with 16 squares. The next step is to determine what alleles each parent can put into their gametes, and the easiest way is to use the FOIL method, which stands for outside, inside, last. So now I'm going to write as so. Now, to fill in the Punnett square, you have to combine the alleles to make the genotype of the offspring as so. For the last step of solving the problem, we're going to have to do an analysis of the genotypes and phenotypes. So first we are going to start by looking for possible genotypes. So what we're going to do is start at the upper left of the Punnett square and make our way down to the lower right of the Punnett square, because that's going to be the easiest way to do it. Now starting with the upper left with the most dominant possible combination, big T, big R, big R. Now that we have the first possible combination, we need to find how many of them there are. In this case, there's only one, so it'd be 6.25%. Making our way down the chart, the next possible combination is big T, big R, little r. And as we can see, there are two of them, so it'd be 12.5%. Now we repeat the process for the rest of the chart.